So we learned about osmosis, where water moves from high concentration to low concentration. But the movement of water in living things is a little more complicated than that. In addition to the amount of water and solute in a solution, we also have to consider pressure. Water potential is a concept that separates out all of these factors and even uses a little bit of math to help us predict which direction water will flow. This is video 6.7 on water potential. We'll cover these objectives, so mark them in your packet or pause to take a closer look. Organizing your notes on this page will sum up almost everything in the presentation on one piece of paper, so consider using this as you go through the video. All right, now we know a little bit about how water moves already, osmosis, right? Where water moves from high concentration to low concentration. But what if pressure was applied? And that does happen in living things. How would pressure affect water movement? Imagine a water balloon. Water's under pressure in the balloon, and if you untied the knot that's holding the water in, where would the water go? Outside the balloon and all over you. And the balloon is an example of how water moves from high pressure to low pressure. And the same thing is true in living things. Now, together, these factors of concentration and pressure make up the concept of water potential. Water potential is abbreviated by the Greek letter psi, which is shown here. And that looks like a letter I with wings, or a letter I saying, I don't know. This mathematical equation sums up the last slide. Water potential, psi, equals pressure potential, psi sub P, plus solute potential, psi sub S. And another thing that's important to remember or to know is that water will move from solutions with high water potential to those with low water potential. Sometimes it's hard to get on board with a concept if we don't know why it matters and why it matters to biology. So one illustrative example of the importance of water potential is in transpiration, the movement of water from the soil to the atmosphere through a plant. Now, in order for water to move from the soil to the roots, to the stem, to the leaves, and then to the atmosphere in transpiration, water potential must be decreasing at each step along the way. And it is, look here. So the water potential in root cells is negative 0.2, and then moving to the stem, the water potential decreases to 0.6, then decreases more to negative 1.5 in the leaves, and then finally negative 100 in the atmosphere. And so with water moving from high water potential to low water potential, water will naturally move from the roots to the stems to the leaves to the atmosphere, and transpiration can happen. Now, here's the simplest case of using this formula to calculate water potentials of two solutions separated by a membrane. For the solution on the left, add up the solute potential and pressure potential, and you get the water potential of 1 bar. Now, bar is the main unit we'll use for water potential. See if you can do the right side. Pause the video while you solve the problem. All right, so adding together the solute potential and pressure potential gives an overall potential of 0 bars. Pretty easy, right? Now, if water flows from high to low water potential, water will flow from the right side, which is higher, to the left side, which is lower. The other major formula we'll use is for solute potential, the contribution of solutes to water potential of a solution. Now, the formula for solute potential is that solute potential equals negative ICRT, and I is the ionization constant, which means how many particles does the solute break up into. For example, sucrose sugar stays as one piece in water, so its ionization constant is 1. Sodium chloride, when dissolved in water, breaks into 2 pieces, 1 sodium ion and 1 chloride ion, so its ionization constant is 2. Now, C is the molar concentration of the solute in the solution, and R is something called the pressure constant. So as a constant, the value of this will always be the same and will always be 0.0831. Now, remember, this equation is given to you on your formula sheet, so you don't have to memorize the equation and you don't have to memorize this number either. And finally, T is for temperature, but the weird thing is that temperature is in Kelvin for this equation to work, which is the Celsius temperature plus 273. So use this equation to try this problem. Calculate the water potential of a 0.5 molar sucrose solution that is in an open container at 37 degrees Celsius. Figure it out, pause, and then start the video up again when you have your answer. If you take control of the information and sort out the variables, you'll see that the ionization constant is 1, the concentration is 0.5 molar, R is a constant number, always 0.0831, and a Celsius temperature of 37 degrees converts to 310 Kelvin. Plug in all of these numbers to our equation, and don't forget that negative at the front. Multiply all those things together, and you'll get negative 12.88 bars. In addition to knowing how to use those two equations, there are some simple rules that you need to know to navigate water potential. And the first rule is that water moves from solutions of high water potential to solutions of lower water potential. And let's check out these two examples to apply the rule. In the system on the left, the water potential of the beaker solution is 0 bars, and the water potential of the cell is negative 0.8 bars. So where will the water move? Since water moves from high water potential to low water potential, water is going to move from the 0 bar beaker to the negative 0.8 bar cell, so into the cell. And as a result, this blood cell will increase in its size and then eventually lyse. Now, what about the situation with the plant cell on the right? Pause the video to figure it out and then hit play to see if you're correct. In this plant cell, negative 6 bars in the cell is higher than negative 1.2 bars outside, so water will flow out of the cell. This plant cell would plasmolyze. Rule number 2 is that pure water has a water potential of 0 bars. What does this mean from a practical standpoint? Well, first, cells would always be hypertonic to pure water, or you could flip that around and say pure water is always hypotonic to cells. And what this means is that animal cells will always lyse in pure water because animal cells will take on that water. Water will flow into the cell until the cell lyses. Water will also flow into the plant cells, but when plant cells are put into pure water, but since plant cells have this cell wall, the plant cells will just become turgid instead of lysing in pure water. Next is rule number 3, pressure increases water potential. So this means that when we combine this rule with the last rule, it means that pressurized pure water will have a positive water potential, a water potential of greater than 0. We should also be able to figure out that animal cells cannot be pressurized because otherwise they would lyse. So the pressure potential of an animal cell will always be 0 bars. And finally, in rule number 3, a turgid plant cell, a plant that's so full of water that the cell is exerting pressure against its cell wall, will have a pressure potential that's positive, a pressure potential that's greater than 0 bars. Moving on to rule number 4, adding solute to a solution decreases water potential. And you can see the calculations in the beakers at the bottom. Using that equation, solute potential equals negative ICRT. Now, assuming that the temperature for all three is a constant 20 degrees Celsius, increasing the solute potential from 0.1 molar to 0.5 to 1.0 decreases the water potential each time, from negative 2.4 to negative 12.2 to negative 24.3 in these examples. This means that pure water with any amount of solute is going to have a negative water potential, a water potential less than 0 bars. And the more solute in a solution means that the solute potential will always get more and more negative with more solute. We're cruising right along here at Rule 5. Rule 5 is that open containers have no pressure, a pressure potential of 0 bars. So if a container is open, like these flasks or a beaker, that pressure potential is going to be 0 bars. Next, pure water in an open container would have a water potential of 0 bars. And that's because there's no solute potential in pure water and there would be no pressure potential. Next, adding any amount of solute will definitely give a negative water potential because pressure potential would be 0 and there's no positive pressure potential to offset that negative solute potential value. Now, the last rule is specific to plant cells and just emphasizes something that we've touched on in the other rules. This rule is that turgid plant cells are pressurized with a positive pressure potential, and normal or flaccid or plasmolyzed plant cells are not pressurized, so this means that their pressure potential is equal to 0 bars. So thinking about this in terms of the other rules as well, is that a normal, flaccid, or plasmolized plant cells will always have a negative water potential value. And that's because pressure potential is 0, so there's no pressure potential to offset the solute potential, which would be negative, with the dissolved solute in the plant cell cytoplasm. And finally, turgid cells may have a negative or positive water potential depending on the solute concentration. Is the turgid plant cell pressurized enough to offset that negative solute potential? Or is it just a little bit pressurized and the negative value of the solute potential is greater than the positive value of the pressure potential? Now, I know that was a lot, and here's the thing. This video is just laying the groundwork for the practice we'll do in class. So there's going to be plenty of time for practice and plenty of time for questions. So let's recap what you need to know right now about water potential. First, water potential is a property of solutions that helps us figure out how water will move, which is always from high to low water potential. Pure water has a water potential of 0, but adding pressure increases the water potential and adding solute decreases the water potential. And finally, we can use math to determine more precisely the water potential of solutions. Then those numbers can be used to predict where the water will flow.